Hello, everybody. My name is Josephine. I'm very excited to be here today with everybody and with everyone, and very happy to be celebrating the International Day of Women and Girls in Science with you. So this session is kicking off an event that consists of 3 2-hour sessions with a program full of very talented and influential women that contribute to forest health today. So if you have not heard of IUFRO before, let me tell you about the International Union of Forest Research Organizations. It is a non-profit, non-governmental global network for forest science cooperation. It unites 15,000 scientists from around 630 member organizations from about 115 different countries. Science within IUFRO is divided into divisions. That includes Division 7 that focuses on forest health, and that is organizing this event today. And within Division 7, we then have 2 research groups, which are pathology and entomology, which are then further divided into working parties that focus on many different aspects of forest health. So how can you get involved with IUFRO? Well, you can directly contact our office holders that work on topics that interest you and that you work on. You can participate in IUFRO co-sponsored events such as conferences, meetings, and webinars like today. You can read and contribute to IUFRO publications, communicate and publish news in IUFRO media, and you can even become a member and eventually become an office holder. So the big event for us, Division 7, and for IUFRO as a whole this year is the IUFRO World Congress that will happen in Stockholm in June 2024. And Division 7 office holders are actually organizing 15 sessions this year. So this is quite a busy event for everybody. Let me take the time to thank our organization committee. I would like to thank Irene Barnes, who will be hosting and moderating session 3 from South Africa, Quentin Kinya, who will be moderating session 2, and that is also taking care of IT for us today, and Jeremy Allison, who has provided support and ideas since the beginning of this project. And I would like to give a special thank you to Prabina Sharma. She is a GIS technician at Clemson University in the USA. Her skills, time, ideas, motivation, and undying enthusiasm has been sincerely appreciated when organizing this event. She has been a fantastic team player. So Prabina will be our moderator for today and will also present her work during session 2. A few housekeeping items. Our moderators, so for this session, this is Prabina, will leave her camera on, so it feels more lively for our speakers, but I would like to ask everyone else to keep your cameras off, so to preserve bandwidth. You are welcome to use the Q&A section to ask questions to our speakers, and Prabina will ask them to the speakers on your behalf. You are welcome to use the chat for any other form of communication. You're welcome to say hi to us, maybe let us know from where you're joining us. If you have any connectivity issues, you notice that the sound or the video is not good, you can also let us know through the chat. As you entered the Zoom call, you might also have noticed that you can choose which room to join. This is because we're using a break room system today. So as our speakers finish with giving their presentation, they will answer a question from the audience for about 2 to 3 minutes. And then as the new speaker comes in, they will join the break room. And the break room is for you to meet our speakers in person or have a one-on-one conversation with the speakers, ask them further questions, and introduce yourself. And then you're welcome to go back and forth between the main room and the break room as much as you want. If you're worried that you will be missing some of the fascinating talks we have today, do not worry. The meeting is being recorded. This will be posted on YouTube afterwards. And we will send you the link to to the YouTube for YouTube after the event. Before we start, I would like to reflect on why we're here and why today is important. So today we're here to celebrate the International Day of Women and Girls in Science. We're here to celebrate the women that have come before us and empower the women that will come after us. And before we hear from our speaker, let me acknowledge why we're here today. So first of all, I would like us to remember that we cannot fight for women's rights without fighting for the rights of all women. And by that, I mean that while we fight against sexism, we must also fight against other systems of oppression that oppress women of color, indigenous women, disabled women, and members of the LGBTQ+ community. Second, our community, which is the environmental sciences community, is approaching parity. And in many instances, women might even outnumber men. But we must keep in mind that representation is not the end game. It is only a stepping stone towards a scientific community that is shaped by a diverse community of people and that serves a diverse community of people. It is a community within which every voice is equally respected and valued. Finally, given the current events, I would also like to acknowledge the women that are not in the room. I am thinking about the millions of women and girls across the globe that are being robbed of their basic human rights, including education, because of the occupation of their homeland, human trafficking, child labor, child marriage, and systemic racism. So let me end this introduction with a quote from the Black lesbian activist and poet Audre Lorde, when she said, "I am not free while any woman is unfree, even when her shackles are very different from my own." And with that, I wish you a happy International Day of Women and Girls in Science. And I will let Pravina take over and introduce our first speaker.
Hi, welcome everyone to the webinar. And with that, I would like to introduce our first speaker of today. And before starting, I would like to apologize if I mispronounce someone's name or something like that. So first speaker for today is Sigrid Netherer. She is a senior scientist and lecturer at the University of Institute of Forest Entomology, Forest Pathology, and Forest Protection at the University of Natural Resources and Life Science, Vienna. From 2018 to 2022, she held an Ellis Reacher Fellowship for highly qualified female senior postdoctoral researcher in Austria, with a main focus on the interactions between European spruce bark beetles, Apis typhogaris, their associated fungi, and Norway spruce under drought. Since her PhD in 2003, Sigrid has been working on forest insect risk assessment, including studies on the pine processionary moth, the impact of global change on the forest insects, and in particular, spruce bark beetle development and outbreak probability. Only recently, Sigrid changed from deputy to coordinator of IUFRO working party on that deals with behavioral and chemical ecology of the forest insect. So she will be presenting today on the role of drought for attack of Norway spurs by the bark beetle Apis typhogaris, field studies and modeling.
Hi, Sigrid, we can't hear you.
Yes, sorry for that. I was for some reason kicked out of the meeting, so I will try to share it screen again. I don't know what happened. told me.
Okay, sorry about that.
But now you see my screen.
Yes.
Right? So that's cool. Oh, again?
Yes, we can see your screen. No, for some reason we can't see.
I don't know what's going on because it always tells me Zoom is Zoom finished. Now you see me.
Yes.
Yes.
And you hear me.
Yeah.
So I really hope that now without any complications we can continue. So thank you very much for inviting me to this exciting event. I'm really happy to present some of my study results here. Today we'll tell you a little bit about the role of drought for the attack of Norway spruce, a very important tree species in Europe, by the bark beetle Ipstypographus, which is the European or Eurasian spruce bark beetle and one of the most aggressive phloem feeders worldwide. So you might also know Dendroctonus ponderosa in North America. This is kind of an equivalent, it has a little bit different life cycle, but it's almost as aggressive as the mountain pine beetle in Europe. So we'll tell you a little bit about field studies I did and some modeling studies. But before, if we think about the life cycle of Ipstypographus, we have to do this on different time scales and also on different spatial scales. So if you think about the time scales, there are phases of endemic population densities, which means that the beetle is present only in very low population densities. While during the population build-up phase, these densities increase, and we finally reach the outbreak phase where there is mass attack and there are a lot of beetles around. Looking at the spatial scales, the beetles, after overwintering, disperse in the landscape and look for adequate stands where there are a lot of host trees, and they're guided during the landscape mainly by olfactory cues, but also by visual cues. Then they finally land on the potentially suitable host trees and again have to decide based on olfactory, gustatory, and probably also haptic cues whether they would like to attack this tree or not. So it can happen, there can happen two things: either they bore into the tree or they leave the tree again because they decide it's not suitable. But if it's suitable and a lot of pioneer-made beetles start boring into the tree, they send out pheromones and lure conspecific female and male beetles, which finally leads to a mass attack. So I would first focus a little bit on the factors triggering such mass outbreaks on landscape and stand scale. And for this purpose, I would like to show you some results of modeling studies I was involved in. And the one really interesting study was conducted on European scales and looked at different factors triggering these outbreaks, like temperature, rainfall, and also wind throw. And we looked at the times at 17 different time series from 1990 to 2010, derived from 8 European countries. And the most northern countries were, for instance, Sweden and Norway, and the most southern countries, so the data points from the most southern areas were lying in France, Italy, Switzerland, and Austria. And based on these time series, which concerned volume of timber lost due to storm damage and also due to spruce bark beetle damage and also climate data, so we used linear mixed effect models and could show that the rainfall of the previous year, but also the rainfall of the current year, had a strong negative effect on the volume of timber lost by attack by due to attack by Ipstypographus. While on the other hand, the temperature conditions of the current year, which means how many generations the spruce bark beetle can potentially develop, and also the storm damage of the previous year, which means how much lying logs will be available for colonization. So these two parameters were significantly and positively affecting the amount of timber lost by Ipstypographus. And we also looked at the interactions between different parameters. And here we found really interesting results, because if you look on the left-hand side of the figures, you see there are the effects of low or smaller scale storm events of the previous years and how they interact with either temperature and or rain. And when I talk about low storm events, I mean that this is more scattered storm events that happen over the landscape. So you have different little patches of storm-thrown trees, and around these patches, so in these patches, spruce bark beetles can develop in lying logs, and then they can easily jump over to standing trees. And you see there was a strong positive interaction between these smaller scale storm events of the previous year and the current summer temperatures. But when it was cool and rainy in the current year after small storm events, there was an antagonistic effect. And even more interesting, if you look at high or large-scale storm events, we did not find these interactions, which means that if there is a large area of fallen or broken trees, beetles have a lot of opportunities to develop in these lying or broken trees, and it takes longer till they jump over to the standing trees. Now I would like to zoom a little bit into one area of one European area, which is lying in Austria. And you see here the circle where I looked at data which I got from the Austrian Federal Forests, and it concerns areas that were adjacent to the Czech Republic. At the time period I focused on was 2015 to 2016, which was exactly the time when these extremely large outbreaks started in Central Europe, which were peaking in 2018, 2019, but which are still going on and destroying a lot of Novi spruce forest. So when I looked at forest stands that were generally highly predisposed to Ipsthypographus because they had a high share of Novi spruce, they had higher stand age, and they were either very dense, which leads to competition between trees, or they were very sparse, which makes these forest stands warmer due to high solar irradiation. So if I looked at these stands and I looked at the probability of attack by Ipsthypographus, so new attack, I found that areas where there were higher effective temperature sums, which means beetles could develop faster and could establish more generations, and areas that were affected by drought, which means that there was transpiration deficits in the summer. So these areas also had a higher probability of new infestations by Ipsthypographus. And even more or stronger was the relationship for infestation that was continued from the previous year, which means the population densities were already quite high, and then warmer temperatures and drier periods again increased the volume loss due to Ipsthypographus. So this brings me to my second question: how does drought now actually affect spruce susceptibility to bark beetle attack? What are the relationships? And if you look at this figure, you see if there is a stressor like drought, but it could also be UV or heat, the conifers such as Novi spruce react quite quickly by closing the stomata, which leads to a disbalance between photosynthesis and respiration. So trees stop their photosynthesis and in this way reduce the production of non-structural carbohydrates, which also means that allocation of these non-structural carbohydrates to either primary or secondary metabolism is changed. And again, this means that under a drought situation, the production of specialized metabolites might either increase, so trees might either invest more into their defense, or in the long run, under a heavy drought, it might even decrease. And specialized metabolites that are really important in bark beetle defense are terpenes, like mono, sesqui, or especially diterpenes, phenolic substances, alkaloids, or also proteins. And then again, in this drought situation, there comes the next stressor, a biotic stressor, the bark beetle Dipteroprofus, which carries exosymbiotic fungi that can also colonize the tree and also add to a depletion of these defense compounds. To have a closer look on these interactions between trees, bark beetles, and their associated fungi, me and my group, we conducted 2 quite comprehensive field studies. The first one was run from 2012 to 2013, and we mainly looked at tree physiological parameters. The second study, so the follow-up study, was run recently from 2019 to 2020, and here we led a focus on associated fungi and also on the chemical defense of the trees. We measured a lot of different parameters, which I cannot all explain here, but a very important one was resin flow, which is one of the main traits of conifer trees to defend bark beetle attack. And we also looked at the reactions of the trees upon inoculation of some species of these associated fungi. Which can, so some of these species can, for instance, establish blue stain in the sapwood. But what trees always do in reaction to this fungi is to establish hypersensitive wound reaction zones. And upon the size of which you can kind of evaluate how strong the defense against these fungi of the trees is. For our study, we developed a method for field bioassays, the so-called attack box bioassays, for which we use these boxes that we mounted to the trees at about 5 or 6 meter height. We put beetles into these boxes and let them be inside for 24 hours. After 24 hours, we removed the boxes and evaluated whether there were successful borings. So, which means that we found a nice boring hole, dry frass, or the borings might also have been defended. Then it was quite clearly visible by these resin flow, quite strong resin flow in reaction to the borings. What we also did in laboratory was heterogeneous arena choice experiments. It's quite a simple method where you offer the beetles 2 different samples from, in our case, from a drought-stressed tree and a non-stressed tree, and beetles could, within a certain period of time, it was 60 minutes, decide where to go. So if they stayed with the sample for 2 minutes, then they had chosen those samples. So briefly, my results, or some of the results. In the first experiment, we found that the roofed trees, so the stressed trees, had much lower resin flow rates than the control trees, or we also had partially roofed trees. This was quite significant. In the second study, then, when we looked at different tree traits in relationship with the host acceptance, or in general with bark beetle tree interactions, we found that trees with higher maximum resin flow rates had much lower attacks, so there were much less interactions between trees and bark beetles at high resin flow. Trees with high resin flow capabilities. In the Petri dish arena experiments, we saw that the beetles preferred trees that were samples of trees that were more stressed. So the black dots you see, these were all the stressed trees. So these were the preferred samples from trees with more negative breed-on-twig water potential, and they also stayed longer near or close to samples, so they liked them more. Independent from the chemical defense, we found in the field bioassays that beetles chose or preferred trees that were less defended by mono and diterpenes. So this was quite a clear relationship. So they just did not try to bore into trees with high monoterpene and diterpene concentrations. We inoculated 2 two fungal species. One was Endoconidiophorae polonica. It's a fungal associate that mainly grows into the sapwood, and Grossmannia penicillata, which prefers growing in the phloem. And when we compared the roofed and the control trees, we saw that the hypersensitive wound reaction zones were much larger at the roof trees for Grossmannia penicillata, which means the trees needed much more time to stop the growth of this fungi, so were less defended against this fungus. We measured bark compounds close to the inoculation holes and also below them. And there were several results we found from these measurements. First, drought alone did not influence the monoterpene contents, but when there were fungi present, the trees accumulated monoterpenes quite significantly, but there was no difference between drought-stressed and controlled trees. However, when we looked at the diterpenes, we saw that the stressed trees accumulated much more diterpenes compared to the controlled trees. So the combination of drought and fungi led to a higher accumulation of these quite important defense compounds. So to summarize these couple of results, I can say that the main triggers of Ipsterographus outbreaks in Europe are wind events, and particularly the scattered wind events, and it's in the lowland, it's mainly drought. Severe drought can strongly reduce resin flow, an important trait against attacks, and impaired resin flow capacities can increase host acceptance of Ipsterographus. However, if drought is only mild or short-term, it does not immediately affect mono and diterpene concentrations. However, the constitutive concentrations of these bark compounds clearly affects the primary host selection by bioinhibitors. What we also saw was that drought-stressed trees are more susceptible to associated fungi, especially what concerns Grossmannia penicillata, and these fungi can further trigger the accumulation of mono and diterpenes. To summarize or to conclude, I can say that it's really important to consider different trophic levels when looking at tree susceptibility to bark beetle attack. So it's not enough to only look at the beetles and the trees. We also have to look at the associated microorganisms like fungi and probably also bacteria. I thank you very much for your attention, and I thank all my cooperators which made such studies possible. And I'm happy to answer questions if there are any.
Thank you, Sigrid, for the wonderful presentation. I cannot see any questions in the chat, but if anyone has question, they are free to turn on their mic and ask to Sigrid at this time.
Can you hear me?
Yes.
Yeah, I have a question for you. I was wondering what was your success rate when you've put this box on the tree with the beetle? Because I know how doing some behavioral work can sometimes be difficult because these beetle never do what you want them to do. So how did it work? Like,
we did not expect a very high success rate because we know that only a few beetles try to attack the tree once they are landing on the tree. So it was over the whole season for trees that were stronger attacked. It was maybe a maximum of 15 borings or so, and some of the trees they really in the first year, for some of the trees, there were no attacks at all. But in the second year, there were attacks at all trees. But yeah, some of them encountered only really few borings, which we expected. So because you have to imagine if it would be so easy for beetles to attack the trees, the outbreaks would, I mean, there wouldn't be any Norway spruce at all remaining, I would say. And what we found in these experiments is that the constitutive defense is important in this very beginning, initial phase of an outbreak. So when beetles land and they decide about boring or not. Later on, it's mainly, I mean, when so many beetles are lured to the trees by the pheromones of the insect itself, it does not play a role anymore. And in between, it's also known that the induced defense is really important. So trees that can better induce the production of defensive compounds, they can defend beetles for a longer time. Yeah, but in this very beginning, it's really the constitutive defense.
I have a question myself as well, because when we were doing the bark beetle study here in America, so the apes apes have the tendency to spread like more quickly compared to the other bark beetles. So I would like to know if you have like found so far like how quickly this species spread. By that I mean like here they can be like the tree are green now and then overnight they can be all brown. So I would just like to know a little bit about
Yeah, this can happen quite fast actually. If a tree is mass attacked, can happen within a few weeks that the tree is completely colonized. So given after the overwintering period, and then there's the swarming time, beetles identify trees and yeah, think it's suitable for colonization and learn their conspecifics. It can happen in the course of really a few weeks that it's completely colonized and dying. Sometimes you realize infestation a little bit later because the canopy can remain green for quite a while. And you might see only in the next year that needles are falling or getting brown. But the tree is already dead and beetles normally already have left the tree. So yes, it's really a fast development, not a slow one, which makes it also so difficult to early detect infestations and to react to these infestations. It's quite a challenge.
Okay, thank you. So if we have no other questions, we would like to move on.
I would like to add another question,
Okay.
if possible. I just wondered if you had ever had the chance to look at the nutritional state of the trees. So could you make a difference between poor soils and rich soils?
This is a very good question. There are some older studies where they tried to look at this, but they did not really find the difference. But it was more the differences between different management regimes, like intensive forest management in pure Norway spruce stands compared to mixed forests. And they did not find clearly significant differences. What I did, I looked at the carbohydrates. I did not show here the results because it would have gone too far. But in such a short time, also regarding the drought effects, we did not see a clear difference in the carbohydrates. It's more that they might even increase in the bark in the first, in reaction to mild or to short-term drought. On the longer run, we would expect, after this first increase, we would expect a decrease in the carbohydrates. And I agree, it might also be really intriguing to look at the nutrients status of the tissue according to differently well-supplied or nutrient-supplied stands. And actually, I can tell that I'm not just now starting another project together with people from SLU, where we also would like to look at the mycorrhizal community, and we would like to compare better and worse water supplied stands, how this acts upon the mycorrhizal community, and how this finally influences infestation of the trees by bark beetles. So I think it was a very good question, very interesting one, that this has not yet been explored so intensively. So there are still a lot of things to do in this direction.
Okay, thank you, Sigrid. I would like
Thanks a lot.
request everyone to ask, like, if they have further questions, to join the break room with Sigrid and then discuss more in detail. Now, considering the time, I would like to move on to our next next speaker of the webinar. So, our next speaker is Dr. Victoria Lantschner. She would Victoria is a biologist and doctor in agronomy. She is a researcher at the Institute of Forestry and Agricultural Research in Argentina. Her research interests focus on describing pattern and understanding population mechanism involved during the stages of the invasion of the forest insects at different spatial scales. Currently, she is studying the population dynamics and invasion ecology of bark and ambrosia beetles. She is handling editor of the scientific journal Neobiota and deputy coordinator of the IUFRO's working party that deals with alien invasive species and international trade. Today, Victoria would be presenting on predicting the distribution of forest insects under future climate scenarios. That is the case study of the South American ambrosia beetles. I would like to request her to start her presentation.
Okay, hi everybody, and thank you, Carolina, for the presentation, and I'm very happy to be here. So, as you may already know, in the last century, the different components of global change, such as climate change, land use transformation, and the globalization of transportation networks, are changing the distribution of species in general and of harmful species in particular. So consequently, it is increasingly important to predict where species will be able to establish and spread in the future. In this study, I will focus on the ambrosia beetle, Megaplatypus mutatus. This species, this is a species from the subfamily Platypodinae, and like other ambrosia beetles, Megaplatypus spores into the trees, creating galleries within the wood. They lay their eggs in galleries where the larvae develop. Within these galleries, they introduce a fungal symbiont that grows and serves as their primary food source. And once the development is complete, the adults emerge and disperse to colonize other trees. And unlike most ambrosia beetles that usually attack dead trees, Megaplatypus is able to attack vigorous trees. This species is very polyphagous. It's able to attack a wide range of trees, mainly broadleaf species. And it is particularly considered a serious pest in commercial tree plantations, especially of poplars and eucalyptus. Oh, sorry. So as you can see in this picture, the galleries degrade the lumber, they weaken the tree stem, and it often then breaks during windstorms, resulting in tree mortality. Well, Megaplatypus mutatus is native from South America, as you can see in this map. It's mainly distributed in northern Argentina, Uruguay, southern Brazil, Paraguay, and Bolivia. And it also has a great invasive potential because it can be accidentally transported in wood products and wood packaging materials used in international trade. In this sense, in 2000, population of this species were found to be established outside of its native range in Italy. So its invasion potential, combined with its wide distribution in South America, as well as its damage to forest plantations, have raised concerns regarding its potential to become a global pest of poplars, eucalypts, and other tree species. So the aim of the study was to determine the potential distribution of Megaplatypus mutatus at a global scale under current and future climatic conditions. To estimate the potential distribution, we collected a database of occurrences of Megaplatypus mutatus within its native range, and we obtained this information from published sources as well as from expert surveys. We also obtained bioclimatic variables related with temperature and precipitation that are expected to determine its distribution. And these variables were obtained from World Climate Database. And then we use this information to perform species distribution modeling using Maxent. We projected the potential distribution of Megaplatypus under different climatic conditions. First, we estimated distribution under current conditions based on historic data from between 1970 to 2000. And then we projected the distribution under future conditions. For that, we used future scenarios based on the shared socioeconomic pathways, the SSPs, and we choose two scenarios. The most optimistic one, the SSP 126, and the most pessimistic one regarding to the future conditions, future climate change, the SSP 585. And we also considered two scenarios in two time periods, a nearer one from 2041 to 2060 and a farther one from 2061 to 2080. And for each SSP, we use the estimations based on 4 different global circulation models. And these ones I'm listing here. And then we average all the results of all the models to have one model. So these are the results of the model under current climatic conditions. This model predicted that Megaplatypus would find suitable conditions to establish in North America, mostly in the United States, in the Mediterranean region, from Europe, Northern Africa, and West Asia, the Southeast Asia, as well as the southern part of South Africa and Australia. So, as you can see here, the model also correctly predicted the suitable conditions for the establishment in Italy, where Megaplatypus is currently established. And these are the predicted distributions under future climatic conditions according to the SSP 126, the most optimistic scenario. So on the top map shows the short-term predictions from 2041 to 2060, and in the bottom, the long-term prediction from 2061 to 2008. In red, you can see the suitable areas predicted under the current condition model that would remain constant in the future. In yellow, the areas that are currently suitable but are predicted to be unsuitable in the future, and in green, the areas that are currently unsuitable but are predicted to be suitable in the future. And you can see that under this scenario, predictions in both periods are very similar, and the areas where expansion is most likely to occur in the future are the forests of northern North America and northern Europe. And these are the predictions under the SSP 585 scenario, the most pessimistic one. Again, the colors indicate change compared to the prediction under current conditions. And you can see that the predictions suggest more abrupt changes under this scenario, and it is expected that megaplatypus will find suitable conditions in a larger area of northern Europe and northern North America, and particularly these changes are more abrupt in the long-term predictions, and it can also be it's predicted to expand also in South and South America. And of course, these models also predict that suitable conditions of the species will decrease in lower latitudes, the yellow parts. So here you can see the global distribution of the hosts that could suffer the most important damage from megaplatypus, the poplars and eucalypts. You can see that the potential distribution of megaplatypus under current climatic conditions largely overlaps with the distribution area of the species. And another significant portion of the distribution of the species could be affected if megaplatypus reaches higher latitudes in the future. When we also identified the relative importance of each bioclimatic variable included in the model in determining the distribution of megaplatypus, we found that the most important variables were the minimum temperature of the coldest month and the maximum temperatures of the warmest month. So, and these are the curves. These show how the predicted probability presence of megaplatypus change as each environmental variables vary. Based on these curves, we can determine that the optimum maximum temperature of for megaplatypus are between 24 and 33 Celsius degrees, and the minimum temperature from -3 to 12 Celsius degrees. And currently, we are also measuring the thermal tolerances of Megaplatypus mutatus in the laboratory. So this is part of Pablo Stefanoni postdoc, who is currently measuring the species maximum and minimum critical temperatures to improve the predictions of these correct correlational models we already did. So these are this is work in progress. We don't have the final results yet, but well, we expect to have them soon. So to summarize, the conclusions of this study are that Megaplatypus mutatus presents suitable conditions to establish in all continents, overlapping with a large part of the global distributions of poplars and eucalypts, and of course other tree species. And it is expected that under climate change conditions, the species distribution will expand towards higher latitudes, especially in North America and Europe. And of course, in order to have more accurate estimation of the probability of invasion of the species, it's also important to estimate the probability of introduction, not only the establishment of the species in the different regions of the world using models such as pathway models. And that's all. Thank you very much for your attention. I would like to thank all the members involved in this project, as well as the institutions that are funding the project. And I also will thank Josephine and the organizing committee for inviting me to participate to this webinar and for the initiative, which I believe is very valuable, and I'm very happy to be part of it.
Thank you, Becky, for the wonderful presentation. Now, I would like to request everyone, if they have any questions, to turn on their mic and ask her.
I believe Maria Rosa has raised her hand. Would you like to?
Yes, I cannot turn on my video, so you don't see my face. It's not a problem. I would like to ask if there are any data relating to the capacity of this beetle to attack oaks.
Oh, I think there's like a very long list of hosts, and I can't remember if oaks are there, but I think yes, they can attack oaks too. I should check it.
Yeah. Just one more question. How far is the biology of this beetle well known? And is there anything on the chemical ecology of this beetle? Any studies?
Yeah, there are studies. I'm not specialist in this issue, but there are some people from parts of Argentina that are studying the chemical ecology. The biology in general is not very well known, but there is some studies. Yeah.
Thank you.
Okay, so because of the time limit, we want to move on to the next speaker. But if you have any more questions for Becky, you can go down below on the right corner and then click on more. And then there is a break room. You can join the break room with Becky and have a discussions if you guys want to take that further. But for now, we would move on to our next speaker, who is Sofía Simeto. Sofía is a PhD candidate currently in the final stages of completing her degree in forest pathology at the University of Minnesota's Department of Plant Pathology under the supervision of Dr. Robert Blanche. Her academic journey began in Uruguay with undergraduate degree on tree endophytes and continued with a master's degree focusing on the variability of native rust fungus affecting native and introduced tree species. Before starting her PhD, Sofía conducted a research on the etiology, biology, and epidemiology of the pathogen in commercial eucalyptus plantations. During her PhD in the United States, she gained experience in researching wood decay fungi and entomopathogenic fungi associated with the emerald ash borer. Sofía also holds a position at Uruguay's National Agricultural Research Institute within the Forestry program, where she conducts research on the forest health of commercial tree plantation. Sofía will be presenting today on entomopathogenic fungi for the control of forest pest experiences and challenges. I would like to request her to start with her presentations.
Hello, everyone, and thank you, Rabina, for this kind introduction. And thank you to the organizing committee of this webinar, which I'm pleased to have been invited to. As Rabina said, I'm going to talk about my experiences researching on biological control with fungi on forest pests. I don't know why it's not moving to the next. Just a second.
You can use your keyboard if you want.
Yeah, I was, but let me try again. Okay. Briefly, before getting into the topic, I would like to show you the, let's say, types of forests I have had the opportunity to work in. Here in Uruguay, as Paulina mentioned, I mainly work in eucalyptus and pine commercial plantations. But while doing my PhD at the University of Minnesota, I had the chance to work on deciduous forests, which were new and fascinating for me. And on all of those different types of forests, I had also the chance to work with biological control for forest pests both in Uruguay and in the United States, but I will be focusing more on my experience in the United States. So what are entomopathogenic fungi? The term is pretty self-explanatory. Entomon refers to insects pathogenic to disease. So these are fungi that parasitize and kill insects. They occur naturally. They control insect populations in nature. And as biological control agents, they are considered a safe alternative, especially to chemical control. So they are a safe alternative to human and animal health, to the environment, and to non-target organisms. They can be included in integrated pest management approaches. And just as a brief reminder, these integrated pest management approaches are supposed to be a package of strategies that combine itself and aim to lower the impact of those pests. So it's always the effect of these biological control agents will always be contributing to the whole. Interestingly enough, some of them have the ability to grow endophytically within the plant or tree host tissue, and that will be could be a potential opportunity for the delivery of those fungi. And there are several commercial products based on entomopathogenic fungi. So I like this very schematic representation of the research towards the development of such biological control agents, where you can see these green arrows represent the several strains. So we usually start with several strains, and we move through different phases of the research, where each of these circles, it's a selection criteria. First, we have this, we select for pathogenicity and virulence, then for their productivity and formulation, then of course for the safety, and those are trials testing if they have an effect, a harmful effect on other organisms, non-target organisms. And at the end, there's of course the production, market, and sales. So on each of these, I like the representation of these stages because they represent kind of a sieve where you, when moving from one to another, you start with several strains, but each of the criteria will remove some of those, and you will end up with just a few. Bearing that in mind, let's move to the entomopathogenic fungi and the emerald ash borer experience I had. So what is emerald ash borer? Agrilus planipennis, is the emerald ash borer, is an invasive wood boring beetle native to northeastern Asia that attacks all native ash species in North America and has had a non-stop spread since the first detection in Michigan, no matter all the quarantine measures and control strategy they try to apply to it. So since its arrival, it has already killed more than 100 million of ash trees. That represents about $10.7 billion in management costs in that period of time, and that is treating, removing, replacing more than 17 million of ash trees. And this cost does not even include the ecological impact of losing ash in natural stands. So no surprise, this is considered one of the most destructive forest pests in North America. So when starting my PhD, I integrated the research team of a big research project aiming to study the fungi associated with the galleries of the emerald ash borer and trying to elucidate the role these groups of fungi play on the infestation and the attack of the beetle. Brief comment here. These compared to for example, bark beetles or ambrosia beetles, especially ambrosia beetles, these are not beetles that had special structures to carry fungal spores like ambrosia beetles. They don't have microangia, so they just we in previous studies and in our study we just saw that fungi are associated with it, but not maybe because they are carrying them naively. So one of the main results of our study, the first part of our study, was that associated to these beetles, we found at least 3 functional guilds, and the entomopathogenic fungi were represented also there. So we ended up with a nice collection of fungal strains of these entomopathogenic fungi associated with the EAB. So here is a brief. So this is the cycle of the beetle, where we have 4 larval instar, which is the most damaging stage of the beetle, and of course the adults, egg adults, and pupae. But once we had this collection of of entomopathogenic fungi, we asked ourselves, can we interfere the EAB cycle at any of those stages? So we try interfering with the adult stage, with the egg stage, and also the larval stage. For a matter of time constraint, I will only show you some of the trials on eggs. So in laboratory, we were able to get fresh EAB eggs from a USDA APHIS facility. We would then cut them individually and put them in these wells from a 24-well plate. We will then produce our fungal spore in water and twin. It was a water and twin fungal spore suspension. And then we would inoculate each of those eggs with 1 droplet of 1 microliter of our fungal suspension. And then we would incubate that at room temperature, and we would have exactly 100 eggs per treatment. So briefly, just some results here. Each of the fungal treatment was repeated at least twice. These are just 2 of them, 2 of the results. The analysis was done through generalized linear mixed effect models. And here in different colors, we see how some of the strains had a significant difference compared to the control in the percentage of unhatched eggs. There to the right, we can see a control. This is a tiny larva emerging from an egg. And sometimes we would also have emergence from eggs hatching, but then the tiny larva, the neonate larva, would produce mycelium and die because of mycosis. So that was also promising in terms of control. So at the end, and this is very briefly about the trial, but at the end of that trial, we ended up with with a table, of summary table of our strains, and we could from there select which were significant, and with those selected strains, we would do further studies, further laboratory studies, or even field trials. As said, I'm just here giving you a glimpse of some of the studies, other trials we did. So we did field trials, spraying the fungal suspension towards the main bowl, and then implanting EAB eggs on healthy ash trees to see if we could target eggs to be laid by females, emulating that. I mean, also we target again by spraying the bulb emerging adults. So right before the emergence was expected, we would do this spraying with fungal spores. We also sprayed also trying to target larvae under the bark, because EAB infested tree the bark tend to crack a lot. So our fungal spore suspension could reach the phloem where the larvae are. And in the laboratory, and bearing in mind the possibility of moving towards an auto-contamination trap, we also did some studies and some preliminary studies by making our fungi to grow on rice. That's the top right picture. Then making adults that were caught alive walk on that, and then they were put together in mesh cage and wait and register death mortality towards the day. And although we have a nice mycose adult there at the bottom, we had some issues here with the control. So I know that individual died because of abovera in that case, but as the control had some high mortality, we could not get a real good result from that one. One field trial I would like to also show you is that the one we did, bearing in mind that these fungi that we have were recovered from healthy tissues, so they were growing there endophytically. So with that in mind, we thought about injecting trees on the root flare. That's the bottom picture there. And then after a certain amount of period of time, we would try to recover the, hopefully, with the hope of recovering our inoculum at different heights. So this was a very, very preliminary study, more kind of a proof of concept study. And we gladly had some nice, although on very low frequencies, we could recover our inoculum up to 4 meters, and in the control, there was no inoculum recovery. And this line of study, it's being continued by another student at the Blanchet lab, but this was the first result that potentially we could use the ability of these fungi to grow endophytically in favor of a possible management control. So all this study and all this effort, I have to say that the field trial results were variable, and some didn't work, as usually happens with field trials. But with control of EAB is certainly not an easy task, but as it is a really important and devastating forest pest, we really believe that every effort is worth doing it. So we were able to build a collection of entomopathogenic fungi strain with varying virulence against different live hosts, and we could even consider that varying virulence to adjust each strain to whatever stage we are aiming to control. The different application methods to deliver these fungi are key, and they are continue to be tested by the students that are now working on this project. And although the biological control, the potential of our isolates might not be a silver bullet, they could we think they could greatly contribute to EAB management within an IPM approach. And as I said, I was only showing you the EAB experience and with the fungi, and this is just a glimpse of another study that we did in Uruguay with the bronzeback, Eucalyptus bronzeback. Some interesting results are reported there, and I just wanted to you to be aware that this is also part of the experience where we can now comment some learned lessons. So forest pest management is for sure a challenging task. The use of native fungal strains, it's an advantage although there are already formulated and commercial products, because native fungal strain will be already adapted to their host environment. That was one example to the Minnesotan winter and the fungi that were already adapted to live there. It's important to consider the whole insect cycle when choosing which insect to target, despite maybe the target it's not the most damaging stage, it's a way to interrupt the cycle. The delivery method is for sure a key aspect, and thinking ahead what type of delivery method you will use can certainly influence the screening criteria. Let's say you might have melanized spores that will survive better under UV lights, or survival structures, or some that produce a lot of spores that would be advantageous for an auto-contamination trap. So it's important to know your fungi, to know the biology of it, and try to think about that and apply it together with the delivery method to plan ahead what type of trials you will be doing. Having a rearing colony of the target insect is for sure very helpful at the moment of planning and doing the laboratory trials, because you can have a batch of insects with the same age, and that you are sure those are not infected from the field. Although, of course, depending on the insect pest, it might be difficult to have a rearing colony. And moving from the laboratory to the field is a huge leap, but it's important to do it, and you have to be prepared to be resilient to frustration, because field trials might fail, but it is important to learn from failure and adapt and adjust, and trying to, if you have nice laboratory results, it's so hard to think that the method to your spores or any propagules to reach the insect, and once they are there, they can for sure go into the cuticle and produce disease. So try to work in multidisciplinary teams. That has helped me a lot. I have had the input from chemical ecologists, from entomologists, from people knowing how to produce inoculums at larger scales, and that has been wonderful. And manage expectations, because entomopathogenic fungi can contribute within IPM programs, but are no silver bullets. And people sometimes expect from microbial control the same results that you would have with chemical control, and that's not the case. So with that, I would like to acknowledge the funding agencies, the university, the Lantschner lab, the Bushley labs, all the collaborators we had in this great project, and also down there the Uruguayan team, and from the Uruguayan side of the bronze bag and entomopathogenic fungi studies. With that, thank you very much, and I would love to answer any question.
Thank you, Sofía, for the great presentation.
I have a question.
Okay.
Hey, Sofía, thank you for a wonderful presentation. So the question is regarding the challenges of working with beneficials in the lab versus the field. So how much real life experience were you able to transition from the lab to the field, and what was your success rate?
Yeah, as I said, when moving to the field, you have to be prepared to deal with failure, because it happens. And for example, we had great results in the lab with these ovicidal effects on eggs. So we thought, okay, let's try on the field spraying the trees according to the timeline where the females would then lay the eggs and hopefully get infected. And for example, we had a very severe drought that summer that just blew our results away. So I think the failure rate is high, but if we don't move from the lab to the field, it just stays in the lab as very interesting results, but it's not what we want. Anyway, that being said, I think it's very, very crucial, if possible, sometimes it's not possible, to think ahead on how you would make your fungal propagules reach your host. So if you are thinking about an auto-contamination trap, then maybe you will select strains that not only are virulent, but also produce a lot of spores in a short period of time. If you are thinking of spores being exposed to environment, harsh conditions, maybe you are looking for harder propagules. And yeah, but for sure, when doing this transition, you have to be aware that failure will be there, but you have to
And
keep trying.
currently, what is your recommendation right now, given your results? Again, limited and number of failures and things that worked and didn't work.
Yeah, well, I think that so many of the trials, the field trials I made are being repeated now by these other students at the lab. My time there ended. I had to return, and I'm writing my dissertation right now. But I think that, for example, the injection of fungi, those were Bovarias, because also those have the smallest spores. So when we injected those trees, we were even wondering, are we going to kill the trees maybe? And we didn't. And we recovered those spores really up high. And this, as I said, was a very, very experimental trial with very low volume of inoculum, because that's the equipment we borrow. So I think that using the ability of those fungi, that are not all of them, but some of them are way reported as being endophytes. So that ability of growing endophytically, I think it's something we have to take advantage from.
Okay, so thank you for the great presentation, and I would like to request everyone else to join the break room for further questions. And moving on to our next speaker, she was the first speaker of our series. So because of some technical difficulties, she have a recorded presentation, and Josephine will be presenting on her behalf, like this recorded presentation. So she is Christine Ouinsavi, a full professor of forestry at University of Parakou, Benin. She leads the Scientific Council of Natural Sciences and Agronomy and Laboratory of Forest Studies and Research, and she coordinates the doctoral training in the natural resource management. She also leads the project funded by International Development Research Center on strengthening gender inclusion in agricultural research for better results in West Africa. She is a member of the Association of Women for Education, Training, and Scientific Research in Benin, and Vice President of Benin National Chapter of Organization for Women for the Developing World. She served as the Minister of Primary Education, Literacy, and National Languages, and as a Minister of Trade. She served as the President of Benin National Commission of UNESCO, and has been awarded by several honorary distinctions. So moving on to her presentation on gender inequalities in higher education and agricultural research, trends and challenges in West Africa. We cannot hear it.
Okay, you tell me you can't hear?
Yeah, it's not audible.
Okay. I'm not too sure why that is. Because my sound isn't. Okay, let me try that again. Yep.
Josephine, it might be because you have to switch the audio on from the computer and not from your mic.
Oh, okay.
You know, and Zoom.
Okay.
So if you go to the mute button,
Yeah.
is that little arrow, and then And then try a different options on your microphone.
Same.
Name assistant, maybe.
No, still no
No.
sound?
We still can't hear.
Okay. Maybe this, sorry about that. No?
No.
Okay. Okay, well, I apologize for that, but I think we should move on to the other speaker. I'll try to fix that problem. Yeah, I'm very sorry for that.
No problem. Let's move on to our next speaker of the series, Dr. Michelle Schrӧder. She is a researcher at the Forestry and Agricultural Biotechnology Institute, University of Pretoria. She completed her PhD in Department of Zoology and Entomology at University of Pretoria, after which she was appointed as a postdoc fellow in the Department of Zoology and Entomology in Forestry and Agricultural Biotechnology Institute, where she is currently appointed as a full-time researcher and co-leads the remote sensing in plant health satellite lab hosted by FABI in collaboration with the ICFR and Ghent University. Her research is focused on insect-plant, insect-insect, and insect-environment interaction, with a focus to develop sustainable and climatically robust insect pest management practices. She will be presenting today on the biological control of Coniferous species in South Africa.
Hi, everyone. Thank you, Pravina, for the introduction. Can you all hear me fine? Yeah.
Yes.
Okay. So, as Pravina mentioned, I am going to talk to you today about the biological control of Coniferous species 2 in South Africa. So, to start with some background information, the Coniferous species 2 is also known as the eucalyptus snout beetle, which was previously known as Coniferous guttulatus. It was originally thought that it was a single species native to Australia that has spread across the world and became invasive and became a very important economically pest for in eucalyptus plantations. It was since discovered that it was actually 3 separate species that forms part of the Coniferous guttulatus species complex that became invasive. Yeah, so the 3 species differ in their invaded distribution, with Coniferous species 2 being present in South Africa. The Coniferous species 2, both the larvae and the adults are defoliating, but it is the larvae that cause most of the damage. The females oviposit their eggs on the new flush of the trees. The larvae emerge and initially feed on the epidermal layer of the leaves, causing these characteristic leaf tracks in the leaves. The older larvae will then consume the entire leaves, which results in defoliation of the crown of the trees, and repeated crown defoliation can result in significant reduction in wood loss over the entire growth period of eucalyptus trees. The biological control program of Coniferous started in 1926 with the introduction of Anopheles natans into South Africa. The egg parasitoid is native to Australia, where it was originally collected in South Australia and Victoria. The program was usually successful, and by 1950, the pest was considered to be under economic control. And this species, the parasitoid was then also distributed to all the other countries where the pest was present at the time. But since this initial success of the program, pest resurgence has occurred, and it is currently still one of the most important pests of eucalyptus in South Africa. And there is thus a definite need to improve the biological control, to strengthen the program, and to get the pest again under economic control. So we started to explore 2 avenues for improving the biological control of Coniferous species 2. The one was increasing the species and genetic diversity of the classical biological control, as well as exploring the use of entomopathogens for managing the pest. So we started our journey knowing that there are some other species of biocontrol agents or natural enemies that do attack the pest, but at that stage, limited information was known about the distribution of the species within the native range. So we started with a survey in the native range in Southeast Australia, collecting coniferous beetles, as well as different life stages to rear parasitoids from the egg capsules and potentially larvae. The insects that was reared out, collected and reared out from the egg capsules were identified. A combination of morphological characteristics and DNA barcoding was used for the coniferous species, and for the parasitoids, it was mainly morphological characteristics used. And subsamples were then sent to relevant taxonomists, Prof. Oberpeer and Gerard Prinsloo, to confirm the identifications. In total, we found 13 species of coniferous within the native range. 5 of the species are from the known cryptic Scutellatus species complex, the previously undescribed species 1, 2, 3, and 4. We also found additional species that could potentially be part of the cryptic species complex, but further taxonomic investigation is required to confirm that. The remainder of the species was not part of the complex. And to focus on, and all of these species varied within their distribution in the native range, but I will from here on focus on the results for coniferous species 2. So looking at the distribution within Australia, we found that it's present throughout the entire range that was surveyed, and the study extended its known distribution further north in Queensland. It's also present in Western Australia. It is introduced into Western Australia, so that's not part of its native range. And another survey by Garcia also found that it is present in Tasmania, but at this stage, it's not clear whether that is part of its native distribution or if it was introduced. The egg parasitoids that was present within the same distribution range was Anaphis nitens, Centrodora domoniidea species, and Clusteroceres species. It's not clear whether Clusteroceres species actually attacks coniferous, as it was only a couple of individuals that was identified from Queensland. The other species, we were able to extend the known distribution range for these species. Anaphis nitens, if you recall, was originally collected from South Australia and Victoria and introduced into South Africa for the biological control agent. And of course, we can't talk about biological control without talking about climate. So the region where it was originally collected has got a Mediterranean climate. And for if we're looking at South Africa, the regions where we are currently seeing outbreaks is more temperate and subtropical climate. So our colleagues in Australia, Sam Wilson, and through BICEP, was able to assist us with climate matching, and we were able to identify Queensland as an area that is got a similar climate to where we are seeing outbreaks in South Africa. So from this work, we were able to then introduce an additionally climatically matched population of Anaphis nitens into South Africa, thereby increasing the genetic diversity. And we have identified Centrodora domoni as a potential species to develop as a biological control agent in South Africa. So we have imported cultures of Centrodora domoni into quarantine, and Idea studied the biology of Centrodora as part of her master's, and she and concluded that it is indeed a good option for us to use as biocontrol. And we have identified some crassomelid species with similar life history in South Africa that we will now pursue host specificity tests to complete the risk assessment of the species before we can consider its safety for release as a biocontrol agent in South Africa. And then to explore the entomopathogens, so we started with looking at the natural diversity of Bavaria that is associated with coniferous species 2 in South Africa, as we have limited knowledge about the native diversity within the eucalyptus plantations. And we evaluated the virulence of the Bavaria species in laboratory trials of the strains that was collected in our plantations. And the map below shows the sampling sites where beetles were collected from which the isolates were made in Mpumalanga, KwaZulu-Natal, and Gauteng. And this was part of Marizan's master's studies. So through molecular techniques, we identified 5 or 6 Bavaria species, Bavaria pseudobassiana, and then 5 species part of the Bavaria bassiana species clade. And 2 of those species, Bavaria neobassiana and Ouinsavi, was recently described to be part of the Bavaria bassiana clade, which is now confirmed to be present in South Africa. And we found 2 novel species of Bavaria as well associated with the Cyniptrus beetles. And we then sub-selected a few strains, 9 strains in total, to do virulence tests in the laboratory. And we found quite a bit of variation in mortality between the different species and strains, with Bavaria namnohensis and one of the novel species showing the highest virulence or highest percentage mortality of infecting the adult beetles. So this really forms the basis for us now to be able to pursue this aspect further, as we now have a clear understanding of the diversity as well as how it influences the beetles. So just in conclusion, the survey that was done in native range really underpins the further development of the classical biological control for Cyniptrus species 2, which led then to 2 additional, or 1 additional species being identified as a potential biocontrol agent, and then also the release of another population of Anopheles natans. And the diversity that we found within the Bavaria species community associated with Cyniptrus species 2 and its virulence really gives us the basis on which to build from to start pursuing this as a potential option for developing this as part of the integrated pest management program for Cyniptrus species 2. Now, there's a lot of people that I would like to thank that have assisted in some way or another along the way with collections in Australia, identifications, and also processing material in the lab, collecting insects for lab cultures and assisting with the rearing in the lab. And then I'd also like to thank the funding bodies and the forestry companies that have assisted in making this study possible. And thank you very much to the organizers for the opportunity to present in this webinar. And thank you for your attention.
Thank you, Dr. Michelle, for the wonderful presentation as well. So if anyone have questions for her.
Yeah.
Okay, yeah, Maria, you can go ahead.
You said that Anopheles nitens worked quite well for many decades until, I don't know, fairly recently. Did you notice, or do you know, if the resurgence of Gonipteros happened all over the place where you have eucalyptus plantations, or if there would be a relationship with the altitude of these plantations? Because Anopheles nitens is known not to be very efficient or not efficient at all above altitudes of about 450, 500 meters.
Yes, so it does. I think climate definitely plays a role. So what we've seen in South Africa, I mean, even historically, you know, when the program was considered to be successful, there were still reports of Cyniptrus outbreaks in spring at high altitudes in South Africa. And that, I think, and that it definitely has to do with the lack of overwintering of the host. So at low winter temperatures, Cyniptrus species, well, the Cyniptrus activity reduces. So there's a very little host material available for the parasitoids to overwinter, which then contributes to the buildup of the Cyniptrus population in spring before the Anopheles population sort of catches up. So there's a bit of a lag time there. But what we are also seeing is outbreaks at low elevation areas, which has not been reported historically. And that is different because historically it was reported that at low altitudes, Cyniptrus has been under economic control and that the parasitoid was very effective. But we do see outbreaks also at the lower altitudes as well, which is a worse subtropical climate in relation to the temperate climate, which is associated with the high altitudes.
Thank you.
Well, thank you, Michelle, again. And we are now moving to our next speaker. We just have 2 more speakers left for this session, and then we have 1 recorded presentation at the end of the session. So our next speaker is Miss Benedicta Swallersberry. I'm sorry if that I mispronounced that, but okay. She is where did that go? Okay, so she is a PhD candidate at the Forestry and Agricultural Research Council based at the University of Pretoria. Her research focuses on enhancing our understanding of phenotypic traits such as growth and virulence in Fusarium succinatum, one of the most important fungal pathogens of pinus species globally. In addition, she also works at the Agriculture Research Council as a research technician and acting curator of the Biodiversity Biobank of South Africa. Her overall research expertise includes microbiology, fungal genomics, and plant health. She strives to advance existing knowledge of important forest and agricultural pathogens by identifying and characterizing genes associated with crucial phenotypic traits. She hopes that through her research contributions, she can aid in unraveling the intricacies of fungal behavior and help keep plants healthy. With that, for today, she will be presenting on the genome-wide association approach to study growth and virulence of Fusarium succinatum. I would like Benedicta to start her presentation.
Okay, thank you very much. Can you all hear me?
Yes, we can hear you fine.
All right, thank you. So thank you for the lovely introduction, and thank you very much for the organizers of this webinar for giving me the opportunity to share my research that I've been doing that is focused on looking at GWAS approach to look at growth and aggressiveness in Fusarium succinatum. So Fusarium succinatum, as you have heard from the introduction, is an important pathogen of pine, and it's globally distributed and is found in America, Europe, Africa, and Asia. It affects pine seedlings and also mature trees, where in mature trees you see this resinous exudates on the trunk of the trees, and then you also see discoloration in seedlings, wilting and dieback, and also damaging of the roots of the seedlings. So because of its importance, a lot of work has gone into studying the genotypes and phenotypes, and also looking at some of the genes that are responsible for phenotypic traits that we see. So some of the ways in which, some of the ways that have been used to look at phenotypic traits and the genes responsible for us seeing these phenotypic traits has relied mostly on genes identified in other Fusarium species. So using a comparative genomic approach, and then also using quantitative trait loci to identify genes. The problem with these approaches is, although we are able to identify genes, it takes a while, because if you look at the comparative genomics approach, it would have to have been studied in another Fusarium species or another fungal species for us to be able to look or characterize this gene. And with QTL studies, that requires for you to have this cross between Fusarium succinatum and another Fusarium isolate, or another Fusarium succinatum isolate and the offspring. And the procedure in getting this offspring is quite laborious. So one of the ways in which we can use to identify genes in fungal species, which has also been used in other species, is a genome-wide association approach. And this approach finds correlations between phenotypes and genotypes. And how it does this is by looking at allele frequency of genetic variants of individuals with similar ancestries, but which display different characteristics. So some would be slower growing and some would be faster growing. So genome-wide association studies have been very successful at identifying candidate genes for different traits in plants, humans, animals, and insects. And much recently, this approach has been used in some fungal species as well. But there's not been any study that has looked at using this approach in Fusarium succinatum. So we wanted to then use this opportunity to get a tool that we can add to our toolbox that would be able to assist us in pinpointing candidate genes that are associated with growth and aggressiveness of Fusarium succinatum. So we were lucky enough because previous studies looking at the diversity of Fusarium succinatum in South Africa had identified a wide genetic diversity of this pathogen across the 5 provinces where pine is planted. So with this data, I then went into looking for potential isolates that I could use to then use a genome-wide association approach. So what we did is we looked at different genotypes from different provinces. Although we couldn't find the same amount of isolates from all the provinces, we were able to get kind of a representation of the genotypic diversity in those provinces, and we managed to get 80 Fusarium succinatum isolates that we could use. So we looked at 2 phenotypic traits, one mycelium growth and aggressiveness. And for mycelial growth, we tested the growth of these isolates at 3 different temperatures, so 20, 25, and 30 degrees Celsius. And after 7 days, we then measured the mycelial growth of these isolates. For aggressiveness, we used a 7-day-old culture where we were able to get a spore suspension in glycerol and inoculate 6-month-old seedlings. So for each isolate, we used 20 seedlings, and this was repeated twice. And then after we start seeing the first death of one of the seedlings or some of the seedlings, we then went to measure the lesion lengths of these the disease. So if we look at the aggressiveness of the population that we had, we thought we would have much variation, but if we look, we see that most of the variation, there is not much variation, between the isolates. However, if we look at Western Cape, Eastern Cape, and KwaZulu-Natal, we do notice that there were a few isolates that showed less aggressiveness on the Pinus patula seedlings that we tested compared to the rest of the population. So this was quite surprising.
Yo,
Vas-y.
je crois que j'ai trouvé ce qui marche pas. Alors, t'entends quelque chose?
Ah bah là, j'entends rien et je vois rien.
Merde. Putain, c'est quoi le problème?
Est-ce que tu as...
Attends. Ce que j'ai fait. Tu vois mon écran là?
Ouais.
Ok.
Women are underrepresented both
T'as le son?
at
Ouais.
the
Ok, super.
Très bien. Ok, j'y retourne.
Ok, merci.
Aggressiveness that we see. And what we saw is when looking at on average, where we had the highest mycelial growth, we saw that there is no correlation between growth of Fusarium succinatum at 25 degrees and the aggressiveness of the pathogen. So now just going into a bit of detail on how we went through our GWAS. So we did whole genome sequencing of all the 80 isolates, and we used the Ion Torrent sequencing platform with 20 times coverage. We then used this data to do a reference alignment with a previously sequenced Fusarium cystinatum isolate, KS17, and then we identified SNPs that we could then use for association testing using SBS Golden Helix. So as we saw with the aggressiveness of the pathogen, there wasn't much variability in terms of the isolates varying. So also what we saw with the genome-wide association, linking the SNPs to that data, we were only able to identify 6 SNPs that were significantly different. However, most of the SNPs were identified at the telomeric ends of the chromosomes. And then only one of the SNP was then identified on chromosome 6 on a gene that when we check the function, it is a lactate dehydrogenase. So this was really interesting because then we wanted to see, are there any other genes within the region that are potentially linked to the SNP that could play a role in Fusarium cystinatum's aggressiveness on Pinus patula. So we identified 3 genes. So the one which the SNP lies on, and then one which encoded a thyrodoxin peroxidase, and also one that encodes a pathogenicity protein. So if we go into the functions and looking at literature and what has been learned from these genes, we find that the lactate dehydrogenase gene has been reported in Candida to be used for adaptation in the intestines of mice. So could this be a role that this gene is playing in Fusarium cystinatum for its adaptation on pine? This we need to further investigate. But moving closer to the Fusarium genomes in Fusarium graminearum, they've shown that the upregulation of this gene is upregulated, in plantar when Fusarium graminearum is used to infect wheat. Also, so the next gene, thyrodoxin peroxidase, has also been reported to be involved in processes such as oxidative stress, heat resistance, stress resistance, virulence, and also the balancing of oxygen, reactive oxygen species in some fungi. And so we would like to explore the functions of these genes in Fusarium cystinatum. Are they playing a similar role in Fusarium cystinatum? So when we move to the putative pathogenicity protein, this gene, I think for me, was the most exciting to find because it's in the name, pathogenicity. But when we looked in literature, there was no published literature on its characterization. So then we did a blast search just to see if there's any other genes out there that have been deposited that might lead us to this, the function of the gene. And then we found one, but it's not published. So it's just deposited in GenBank. And this was in Fusarium oxysporum, and this was from a student's thesis that they had just deposited and not published, I think, which then emphasizes the importance of publishing our research, because then as a community, we're able to then have something to reference. But this gene was particularly interesting also because when we looked at other Fusarium species closely related to Fusarium cystinatum, this being Temperatum and Fusarium pilosicola, we find that this gene is absent. So it is not present in the genomes of Fusarium temperatum or Fusarium pilosicola. Fusarium temperatum is not a pathogen of pine, and pilosicola has been found on pine, but has not been shown to cause disease on pine. So I think it's important for us to then go a step further to understand the role that this gene plays in the pathogenicity of Fusarium cycinatum. And because a study can only go for so long, we had to pass this on to a master's student who has been working on characterizing the function of this gene in Fusarium cycinatum. And she's using a CRISPR-Cas9 approach to do this. So just a summary of some of this work. We have now added this tool that we can use to aid in studying and understanding the molecular basis of some key phenotypic traits in Fusarium cycinatum. And the genes that we've identified offer an opportunity for us to characterize their role and see how and what pathways they're involved in leading up to aggressiveness or any other phenotypic trait of Fusarium cycinatum. And this all will also provide avenues for us to develop better control and management strategies for Fusarium cycinatum. So with that, I'd like to thank the funding bodies that made this research possible, and for IUFRO again for inviting me to share my work. But also it won't do justice if I don't acknowledge all the women who have been involved in this project, some directly, some indirectly, just having conversations with them about how one can go about certain experiments. And thank you, and we do all of this to keep pine trees healthy. Thank you.
Thank you, Benedicta, for the wonderful presentation. Now I would like to request if anyone has any question, they can raise their hand or unmute the mic and ask questions. Okay, so I can see some of the claps, but
Well, I have a question for you, Benedicta. So how do you even start looking for the function of the gene? How do you, like, that's not my field. I have no idea where I would start doing
Okay,
something like that.
So we do have a gene knockout system. So the one that I mentioned is the CRISPR-Cas9 system that Alida is working on. So what you do is we would then knock out the gene and try and test the pathogenicity of a mutant versus a wild type and see how the differences are. Does the mutant cause less aggressiveness on the plant? And then also study other traits like growth. Does it affect the sporulation of the isolate if you knock out the gene? And then from there, we were able to characterize its role. Okay, this gene is involved in growth, in pathogenicity. It affects the sporulation. So the absence of it would affect how sporulation happens. So by knocking out the gene, then we are able to understand, okay, this is what the gene is involved in. And then from there, take it a step further to see how we can then implement a way in which we can use this to control Fusarium cycinatum in field.
So we just have our last recorded presentation for this session, and I would like Josephine to play that.
Hello, everyone. I'm Professor Christine Ouinsavi from University of Parakou in Benin. And I'm presenting on gender inequalities in higher education and agricultural research, talking about trends and challenges in West Africa. Dear participants, gender disparity in STEM are undeniable, and many efforts have focused on comparing statistics and balancing numbers. In Africa, education should be the main response to the reversal of social gender inequalities. In higher education sector, and let's say especially in agronomy, the realities seem deeper and deserve more efforts. In higher education in Africa, as in several socio-professional sectors, women are underrepresented both at the academic and governance levels, also among students, and within the system of conception and use of technologies. But countries' statistics are not always up to date. But considering female application for the latest CABES session, covering higher education institutions in the 19 countries in the CAMES area, we could notice that females are represented at about 4% in engineering science, 8% in mathematics and physical science, 16% in social science, 17 to 18% in economic and natural science, 23% in health science, and 24% in sport activities. In the agricultural field, we easily note that let's say we note the exclusion of women from all stages of the processes, from identifying needs, design, and developing technological innovation. This consequently lead to the exclusion from the benefit of these technologies. The situation leads us to tackle research methodologies in agricultural science for better gender inclusion. That's why the purpose of our research was to measure gender bias and disparities, and to produce new results that change institutional gender policies in higher education and agricultural research. And to do this, we gathered statistics from registration books at universities, and we surveyed training and research unit managers, and we supplemented by individual interviews. We also carried out systematic review on the factors associated with gender inequality in agricultural research methods. We carried out then a quantitative meta-analysis focusing on two sub-objectives, meaning the relationship between the type of sampling and the inclusion of gender in the samples, and then the estimation of the proportion of women in the study samples. Finally, we carried out an experimental case study based on a participatory approach to identify, together with women and men farmers, the gender inequalities in agricultural research factors, in order to see how to formulate an inclusive and gender-sensitive methodology. So as key findings, we noticed that among students, female represent less than 30%, and the trend is even worse when we consider a different level of graduation. But it is important to highlight here that we recorded more loss at doctorate level. This involves the conflict of social condition with education, because at the certain age when female engage in doctorate studies program, you all know that it is a period where biological clocks start in ringing. In Niger, as shown by this figure, the situation was more worrying as females were almost absent in higher education, but a hope still rising from the proportion growing slowly up to about 30% in 2021. Analyzing the trends of gender asymmetry index, it could be noticed that although this index value is increasing, approaching zero, meaning that we are approaching equality between male and female representation in society, there still be a great imbalance toward women. The trends are the same at university level, and we try to find it out based on some case studies in Benin universities. Talking about representation of women at the head of agricultural training and research institutions, we could find out that 9.52% representation of women is noticed at the head of institution. The gender parity index calculated reveal 0.1 in Benin and about 0.38 in Niger. We also calculated gender, the absolute gender gap, which is 34. This means that a larger gender inequality still exists in favor of men. While analyzing gender inclusion from research methodologies by meta-analysis, we noticed that women's compliance in the study samples was estimated at 32% when we consider fixed effect model, and 26% for a random effect model. So we came out with the fact that the sampling methods and techniques were not significantly associated with overall a proportion of women in the samples of the included studies. We then co-identified the main reasons of gender disparities using a participatory research method and found out that according to farmers of both sexes, the reason of gender inequality in studies are a patriarchal regime, talking about censorship and expression, the nature of some survey, the targets, meaning that some study focus especially on some category because of the type of study. Some examples are given here, like talking about fishing, cattle breeding. You sample only men, but when you are working on net collection, processing of fishery, and so on, the sample is most of the time composed of mainly females. There are level of education, choice of interviewers too, because in a rural area in Africa, in our place, when the interviewer is a male, he hardly has access to female for his interview because of cultural consideration. So with those farmers, we identify some solutions talking about ensuring a good education of a girl at home and also at school, sensitization and training parents and spouses to help girls and women in daily tasks in order to help them having time to focus on research activities. Also, we think that the use of some mathematical equation and formulae is necessary to determine a sample size that is representative of both population and the sex, both sex, for gender inclusion in those samples. From all this, we started some concrete actions based on accentuation of coaching of women, young women in higher education and agricultural research. And from that, we made some training on various topics for research enhancement. We also help for finding scholarship to younger women in agricultural research. We promote also mentoring for younger women scientists, and we created a platform for intergenerational exchange and promotion of mentoring. That platform link is www.agriwomen.org. As a perspective, we intend to install a long-term device for collecting more accurate and continuous data on the evolution of women's participation in agricultural research. Also to collect data on gender inclusion to secondary and technical education. We also intend to extend data collection on gender bias measurement to management of agricultural development projects in West Africa, because the challenge is not only at highest education. We finally think that it is important, and we intend to carry out some political institutional lobby by developing advocacy documents and using it for sensitization of key actors. Talking about those key actors, those key actors involve policy makers and other stakeholders like a parliamentarian and members of the executive board. Also key person like a head of association and gender promotion structures, and then head of some community institution like ECOWAS and UMOA. All in one, we also think that we will develop a network with other teams for more impact on the field. So, dear participants, that's what we prepare, what intend to share with you through this communication. I will finish by thanking IDRC for supporting these research activities in Benin, Togo, and Niger, for providing us with funding. So thank you for your kind attention.
All right, I believe that would be the end of our session, and I would like to thank all of our presenters once again for the wonderful presentations, and all of the participants for being with us so far and having interest in this webinar session.
Thanks for coming, everybody.